One of the most important steps for cleaning up resting state data is denoising. Of course, removing noise is necessary for any imaging analysis, but resting state data, in particular, is very susceptible to artifacts such as linear drifts and motion effects. To begin denoising the data, click the Done button from the Setup tab. A new menu will appear, prompting you to select which resting state analyses you want to do. Any combination of ROI to ROI, seed to voxel, and voxel to voxel. Don't worry now about what these mean. We'll cover them in a later video. All three are selected by default, and we'll leave them as they are for now. Click Start to begin the denoising step. After a few minutes, you will have access to the Denoising tab. The window in the upper left corner shows the confound regressors that are used to account for different sources of noise, while the window on the right shows how much signal can be explained by each regressor. By default, Conn will include regressors derived from the tissue types that you generated in the ROI section of the Setup tab, and the first level covariates of the Setup tab as well. Conn extracts 5 principal components from both the white matter and the CSF ROIs. These best represent the signal profile in those regions. If you click on the string, for example, white matter 5P, below you will see an illustration of each component's time series. In the Confound Dimensions panel, you can change the number of components. For example, try entering a value of 10 and see how the time series panel changes. Although including more components can filter out more of the signal from these regions, you quickly reach a point of diminishing returns. More regressors also means more degrees of freedom, which can reduce the statistical power of your model. For most experiments, the default of 5 components will be enough. Within this panel, you can also choose to add higher order derivatives. If this is done at all, it is usually done to the motion regressors specified here under realignment. For example, first and second order derivatives can capture more subtle movements that are not accounted for by the traditional translations and rotations. Polynomial expansion is a similar concept, adding either a quadratic or a cubic exponent to the regressor, although this is not commonly done. For now, I'm going to reset these back to their defaults. The Filter checkbox specifies whether the regressor should be bandpass filtered before it is entered into the model. You can do this for individual regressors by highlighting the confound regressor and checking the box, or by changing the After Regression RegBP option to Simultaneous. The reasons for choosing to do these are discussed more in the link to the book below. In the Additional Steps panel, you can also choose to add higher order detrending. For example, if you have a very long scan session and you think you may need to model more complex scanner drift. And despiking. Despiking will artificially lower the signal of voxels that are abnormally high, which are not accounted for by high motion. For most data, choosing to either despike or not despike will give similar results. However, for unusually noisy data, despiking can be useful for removing artificially high signal. The right side of the denoising area will automatically update a figure showing how the regressors load onto the resting state data. The distributions over here show the effect of your denoising regressors and parameters both before and after they are applied, with blue being before and yellow being after. In general, the raw resting state data is skewed to generate positive correlations, mostly as a result of confound regressors such as motion. Note how the distribution of connectivity values is centered closer to 0 after denoising is applied. The scatter plot below the distributions shows another view of the same principle. Data that is denoised shows a more uniform distribution in connectivity values as one tests for correlations at voxels farther away from the seed voxel. This figure here is something called a carpet plot, which unravels each volume from a 3-dimensional cube of voxels into a 2-dimensional plot of squares. Each column represents an individual volume, and each row is an individual voxel in that volume. Denoising smooths out the rougher transitions between voxels that are probably caused by motion, scanner drift, and physiological noise, and the time series plot of the global signal reflects this smoothing out as well. Lastly, the preview brain on the right-hand side shows the percent of variance in the bold signal explained by the currently highlighted confound regressor. The loading of the explained variance should correspond to the regressor that is highlighted. For example, without me showing you, can you guess what this confound regressor represents? Do you think it is movement, the white matter components, or the cerebrospinal fluid components, and why? For most datasets, these figures right here should look pretty similar. The distribution of connectivity values will be centered close to 0, and the bold time series will be smoothed out. If the data passes those checks, you are ready to begin estimating a general linear model using those regressors. The next step of our analysis is to create a first-level model using the regressors in the denoising and setup tabs. After regressing out the confound regressors, a correlation value in each voxel is calculated by averaging across the signal in each seed and correlating it with all the other voxels in the brain. To run the first level model, click Done from the Denoising tab. You have options for which type of resting state analyses you want to do. Leave all of the boxes checked for now and click the Start button. The toolbox will take a couple of minutes to denoise the data and generate each of the correlation maps you specified. When it is done, you will have access to the Analyses First Level tab. It automatically opens so that we can review the correlation maps. If you recall from the Setup tab, several different ROIs were generated: gray matter, white matter, and CSF. The latter two primarily for denoising using a NAT comp core, and atlases and networks. The atlases and networks ROIs parcelate the brain into different regions, or seeds. The time series of the gray matter is averaged across all of the voxels in each of the seeds, and correlation maps are generated for both the correlation of that seed to all the other seeds, and of each seed to all of the other voxels in the brain. Clicking on the Seed to Voxel or ROI to ROI buttons will display two distinct areas in the window, similar to the setup in the Denoising tab. The left area, Seed-Based Connectivity Analyses, lists different analysis types and options that you can choose from. The right area, Preview First Level Analysis Results, shows how the average time series of the currently selected seed correlates with other voxels in the brain. Since we are only looking at resting state data, functional connectivity weighted GLM is the most appropriate option to use. Later on, we will see how to do a generalized psychophysiological interaction or GPPI analysis, which can be used with task-related datasets. All that we want to do for now is to validate that the first level correlation maps look okay. In the Seeds Sources panel, down here, highlight different seeds to observe how the connectivity map changes in the Preview First Analysis Results area. What do you notice about it as you select different seeds? Do they match up with what you would predict? For example, frontal pole right should, obviously, correlate the most in the right frontal pole, and frontal pole left should correlate the most in the left frontal pole. Do this for a handful of seeds, making sure that the highest correlation is within the seed itself. You can also check other seeds and networks, such as the default mode network, to make sure that it generates the pattern you would expect. In this case, the default mode will be most correlated between the medial prefrontal cortex ROI and the posterior cingulate gyrus ROI. If you are satisfied with the preview of these correlation maps, click the Done button. This enables the second level results tab. This will only take about a minute to run. Once it finishes, it enables the results second level tab, which, unfortunately at this point, won't be able to give us any useful results with only one subject. We will have to include more subjects in order to run a valid group analysis, which we will cover in the next video.